So welcome everybody to our Ideas Forum. This is the first of the new year and going to be a superb one. We're really very fortunate today to have two wonderful talents with us, and I'm going to introduce them both at the beginning, and then after each one talks, we have questions. So I'm going to start by introducing Dr. Darina Petrovsky. She's an assistant professor at Duke-UNC School of Nursing. Prior to joining Duke in 2024, she was an assistant professor at Rutgers University School of Nursing and also core faculty at their Institute for Healthcare Policy and Aging Research. She also has a bachelor's of musical arts. That unique background, combining the music training and her nursing science, positioned her to launch her research career with improving the lives of older adults living with cognitive impairment using non-pharmacologic approaches. Her additional research interests include ways to achieve health equity among older adults living with cognitive impairment and their caregivers, as well as how music can be used to prevent cognitive decline. So we'll be hearing from her first. Our second speaker is going to be Dr. Silas Buck. He's a postdoctoral associate at the Duke-University School of Medicine in the Department of Neurology. Prior to joining Duke in 2023, he did his PhD in neuroscience at the University of Pittsburgh, where he studied why dopamine neurons may be more vulnerable to degeneration in Parkinson's disease compared to others. He pursued his interest in uncovering fundamental mechanisms underlying neurodegeneration in Parkinson's disease, as well as other neurodegenerative diseases, and he did that in Dr. Laurie Sanders' lab. His current research will help us advance our understanding of tau pathology and certain types of Parkinson's disease, and the goal is to understand why we observe it in Parkinson's with the specific mutations of LRRK2. Not only will this inform treatments for those patients, but also our patients with other tau pathologies, such as Alzheimer's disease and other related dementias. So this looks like two very exciting presentations. So with that, I'd like to ask Dr. Petrovsky to launch us.
Sure. Thank you so much, Dan, for the introduction. Let me just start here. So, I am really excited to be here. So I'll be talking about how to promote well-being of persons living with dementia and their care partners using music-based approaches. This is a picture of actually my grandmother who passed away now more than I think a decade ago, and her favorite cat who traveled with her from Russia to the US. And I took this picture, I think I was on a winter break, and ended up, I submitted it for a competition at the Grants Makers in Aging, and it won a prize. It won like second place in the category of aging in place. So I love to include this as sort of my first slide to keep me grounded and keep me excited about the work that I'm doing. So I wanted, in my about 20 minutes that I have, I want to share a little bit more about my trajectory into the field of music and dementia, spend the bulk of the talk describing the different mechanisms of music and memory, and a little bit about other forms of the arts, and end with talking about what are some of the benefits of music-based interventions in this population. So I grew up, as I alluded to, I actually grew up in Russia, immigrated to the States right at the start of high school, and I've always been involved in music, grew up in a very musical family, and took up piano right around the age of 6 or 7. And when I moved to the States to start high school, one of the gigs that I had, one of the sort of paid opportunity that I stumbled upon was actually playing in nursing homes, libraries, so living places. So I played the piano, sometimes by myself, sometimes with other singers, as pictured here. Because I couldn't drive myself, my mom would come and drive me places, and that's her in the background. She would be the designated page turner for these concerts. But it really gave me, back then I didn't think of it as a career important decision. I saw it as a sort of an opportunity to get out, to practice my skills. But now looking back, I definitely see the connection, because I think that's the moment that started my continued my interest in the care of older adults, of them living out in the community, and the connections that they showed me they had with music that I would play. So I thought that was a fantastic opportunity to get involved and really influence my career moving forward. I sort of had a roundabout way of getting to where I am, and I think most of you in the audience may also agree that it's usually sometimes not the path that you think you're going to take. I started out and got my degree, as Jen mentioned, in musical arts at Michigan. And then I had a very influential experience. I shadowed a nurse right at the end of my third year, but my junior year, and found that career. And it seemed to me that this was sort of a perfect combination of an opportunity to provide bedside care and care for older adults, and potentially down the road explore possibility of doing research. So that's kind of what I did. I ended up moving to Cleveland. I did a 2-year master's program, got my nursing license, worked a little bit primarily in nursing homes upon graduation, and at that time got connected to a researcher at Case Western who's done some foundational work at the intersection of music and older adults. She was right at retirement, and so she said, I couldn't really mentor you, but I think you have a future in this area. I would encourage you to pursue research in this area. So I ended up getting my PhD at Penn, and all of my work since my PhD really focused on the intersection of music and the brain and older adults, and I'll speak a little bit about what I have done so far. You may not be a stranger to these type of headlines. People send them quite frequently to me. This is all over NPR, BBC, and other news outlets, the Washington Post. But sort of this notion of the importance of the arts, sometimes using the different kind of arts to inspire creativity or help you focus at doing something. This whole idea of, are musicians really better off than non-musicians in terms of cognitive skills and resilience to adversity, and in the strong and obviously exploring the connection between music and memories. So this has become quite a popular art topic in terms of how to use music, and especially help older adults, which is what has been my focus. There's this notion, and I'll talk a little bit about the research behind it, that we really have a strong connection with music that was prominent and important to us during our sort of formative years, which is considered about to be adolescence and young adulthood. And I think when you think about it, it really makes sense. That's when we really come of age, become young adults, and that music tends to those musical preferences tend to stick with us, although they can also change in the disease process. And as a neuroscientist, I've always been interested in symptom management and using music as a tool to help address those bothersome symptoms. And I'm using sort of the term music interventions, but that can include anything from music listening to an involvement of a music therapist in a music therapy session. And you can also play music, and that can have additional benefits to you as well. So it's a pretty broad term, but it includes different sort of variability of how music can be used. There's been a number of studies that and longitudinally have shown that as adults participate in leisure activities, which includes oftentimes musical activities, but sometimes they're included under leisure activities. There's been a number of research that shows that it's really been associated with decreased risk for dementia and a lower rate of cognitive decline over the years. There's been several studies looking specifically at musical activities. They're a little bit harder to do those studies because longitudinal studies have not included history of musical activities that provides you that longitudinal look. But in some populations, it has been associated with decreased risk for dementia. There's also this notion, the body of literature that's been around for now probably 15, 20 years, that cross-sectionally, when you compared older musicians to non-musicians, which is arbitrarily defined as having less than 10 years of musical experience or more than 10 years, that the older musicians tend to do better on a variety of cognitive tests, as I list here, compared to non-musicians. So there's sort of this notion that by participating in and sort of doing and playing a musical instrument or being involved in music for a longer period of time tend to have some cognitive benefits later in life. But you can imagine that there are limitations with these types of studies. It's hard to compare somebody and arbitrarily define musical experiences as 10 years or less, along with some other limitations as well. So this is an excerpt of the video I would love to show you. I was actually invited to be on a panel just last week. I live in Moore County, and this was in Southern Pines. This movie, the whole in this entirety, is about 70 minutes, was shown to the community, to our community here where I reside, and we had a panel of myself, a couple of local musicians, and somebody living with early onset Alzheimer's disease to talk about the benefits of music. So I love this video. If any of you have seen this, I'm sorry you'll see it again, but this movie came out about now more than probably 10 years ago now. This is some of the work from Dr. Oliver Sacks out in nursing homes in New York City. And the social worker, his name is Dan Cohen, who, and the movie maker, they followed the social worker for about 3 years, capturing these moments of music sort of bring people back to life. So let's see it here.
I have one resident that barely opened her eyes. She didn't respond. As much as I tried, I knew her for 2 years. No matter what I tried, massage wouldn't work, nothing worked. When we got introduced to the iPods and the family told me the things that she liked, it was amazing once we put the iPod on her. She started shaking her feet. She started moving her head. Her son was just amazed. Okay, can we stop? Because now I'm getting all I'm seeing her all over again.
Hi, Papa. Hi, Papa.
Huh?
How you doing?
I'm all right. Fine. Wait.
Who am I?
I don't know. Wait a minute. I don't know.
Okay, it's Cherry. How long has he been in the nursing home? Approximately 10 years. He was having seizures, and my mother couldn't handle him at home. Of course, it affected me greatly because he was always, fun-loving, singing, every occasion he would come out with a song, no matter where he was. I remember as a child, he used to walk us down the street, me and my brother, and he would stop and do Singing in the Rain. He would have us jumping and swinging around poles. He was, he was good. He was always into music, always loved singing, dancing.
His name is Henry Dreyer.
Uh-huh.
And I'm looking more or less for religious music for him.
Okay.
Because he enjoys music and he always quoting the Bible. So I'd rather have that for him.
We first see Henry inert, maybe depressed, unresponsive, and almost unalive.
Henry.
Yeah?
Henry.
Yes, sir.
I found your music. You want your music now?
What do you want me?
Let's try your music, okay? And then you tell me if it's too loud or not.
Then he is given an iPod containing, we know, his favorite music. And immediately he lights up. His face assumes expression. His eyes open wide. He starts to sing and to rock and to move his arms, and he's being animated by the music.
And he used to always sit on the unit with his head like this. He didn't really talk to much people. And then when I introduced the music to him, this is his reaction ever since.
The philosopher Kant once called music the quickening art, and Henry is being quickened. He's being brought to life.
Yeah.
I'm going to take the music for 1 second, okay? Just to ask you a few questions. Okay? I'm going to give it back to you.
Uh-huh.
Okay.
The effect of this doesn't stop, because when the headphones are taken off, Henry, normally mute and virtually unable to answer the simplest yes or no questions, is quite voluble.
Henry,
Yeah.
do you like the iPod? Do you like the music you're hearing?
Yes.
Tell me about your music.
Well, I don't have one, I mean.
Do you like music?
Yeah, I'm crazy about music. You play beautiful music, beautiful sound. Beautiful.
Did you play music when you were? Did you like music when you were young?
Yes, I went to big dances and things.
What was your favorite music when you were young?
Well, I guess, Cab Calloway was my number one band guy I liked.
What was your favorite Cab Calloway song?
Oh, I'll be home there for Christmas. Oh, you can count and plan on me with plenty of snow, mistletoe, present wrapped around your tree. Ow!
So in some sense, Henry is restored to himself. He has remembered who he is, and he's reacquired his identity.
I'm sorry, I'll pause here in the interest of time. I would encourage you, if anybody who are interested in have some downtime, this movie is available. The whole movie is available, I think, on Netflix and Amazon Prime, if I'm not mistaken. But it takes through a series of sort of case studies and follows Dan over, as I mentioned, over 3 years. And Henry's a reaction to music. It is, I wouldn't say it's extremely it's extreme, but it does show sort of this going from very, as you can tell, very mute, kind of non-responsive to being a lot more responsive, and more, very importantly, gives him a chance to connect with a part of himself and that he's maybe lost since he has been in the nursing home. And so this movie really, this clip of Henry went viral right after I think the movie premiered, and has really given a public an opportunity to share their own experiences of their family members and whether living at home or in a nursing home, and how they have connected with music. So whenever I go out and give talks to the public, that's probably number one example that people have used describing sort of how music has impacted their life. And there's only been just a handful of studies that talked about that perhaps try to explain this phenomenon of why individuals with dementia, why is music pretty preserved in those. And so in this particular study, this was now published in 2015, so now almost 9 years ago, they did a two-phase approach where they first had 32 sort of normally healthy younger adults, and they scanned, they had them in the scanner, and they identified areas of their brain that were responsible for long-term musical memory. They had to come in and share with the scientists their playlist of their favorite music, and then while they were listening, they were scanned. And then the second sort of phase of that study, they brought in 20 individuals with Alzheimer's disease. I think these were in the mild stages, and compared to them to 34 cognitively normal, older adults. And what they found is that when they were just looking at the areas that were sort of identified to encode this long-term musical memory, they exhibited lesser cortical atrophy in those areas. So that was one of the first studies to really get at, why do we see this music, long-term memory for music preserved in dementia, in this case, Alzheimer's disease. A couple of years later, there was a group out of the University of Utah, I believe, that had a little smaller sample size. They were not, also did not compare them to cognitively normal older adults. They also had them bring in their personalized music, and unlike the other studies where they looked at specific regions of the brain, they looked at the connectivity between different regions in the brain responsible for music perception and long-term memory. And similar, they also found that those areas didn't exhibit as much atrophy as compared to areas not responsible for music perception. So whenever I talk to the public about the importance of this work and why do we see this preservation of long-term memory, those are the types of studies and citations that I use. And for a long time, and since, I'm very much my work has been focused on the underlying mechanisms and sort of theories behind the connection between music and memories and why music is such a powerful stimulus. And there's been several models, several theories proposed. You can list them here. You can think of that music as not just a social activity, but obviously a strong mentally stimulating activity. Even music listening by itself, which is what my research has focused on, can be a very stimulating activity and can have actually a negative response to music. If for music training specifically, you can think of it as a very highly specialized skill. So anytime you practice a skill over and over again, that requires practice and dedication. So that in itself provides cognitive benefits. And of course, there isn't necessarily one mechanism. There could be many things in place, especially outside of the lab environment. And so I think for every music intervention, there's different things sort of working together. Some of the work that I've done looking at how choirs can be used to promote healthy aging in older adults. This was the work I did with a mentor at UCSF. She had one of the largest studies of about 390 cognitively normal older adults, a relatively diverse sample. And she conducted an RCT, and after 6 months, unfortunately, she did not find benefits for cognition or physical function, but she did find for those who were randomized to the choir group that they experienced a decrease in their loneliness, an increase in the interest of in life. There's many different benefits specifically for music and dementia. Here I list some. My work has focused primarily on sleep and mood and neuropsychiatric symptoms of dementia, and it's really been hard to keep up with the literature in the last 5 years just because of really explosion of work in this area. Here I just provide one example of a study I did as a doctoral student where if you're familiar with PACE programs, Program of All-Inclusive Care for the Elderly, I sort of joined a natural experiment. There was a choir that would frequently get together and sing in one of the PACE programs. This was in Philadelphia, and they were primarily Black older adults, some with cognitive impairments, some not. And I asked them, what is it about this program that keeps you engaged and keeps you coming back? And so I did some qualitative work, and they really described the joy that they had for music, the joy that they had for singing, and they loved how they were able to share their talents, and it sort of gave them purpose as they were meeting. As a postdoctoral fellow, I completed a randomized clinical trial where I looked specifically at music listening to improve symptoms of sleep disruption in older adults with dementia. This was we enrolled about 33 dyads who had either had a diagnosis of dementia or had a self-reported memory problem, which was confirmed with the CDR. And it was primarily feasibility study. I used it as a jumping off point to write my career development award, to show that, and this is just one of the quotes from the study, that older adults really did enjoy listening to music, that they saw that as a way to supplement sort of other methods that they used to help them improve sleep. But technology that I used to deliver music was not very age-friendly or older adult-friendly. And so based on that, I wrote a career development award, which was funded. So I'm in year 4 now of the award. And then in this project, we're building an app called Composer. It's a mobile app. And we're going to look at the acceptability as well as preliminary efficacy of this intervention with the music being delivered on the app for older adults living with dementia and care partners. So I'm sort of in stage 1 right now. The study's been a little bit delayed since I moved institutions and had to find a new software developer company. But we're moving ahead and really excited to do this work. I just have 1 or 2 minutes left. So I want to share a couple of currently active arts-based programs that are available for older adults, because I'm always asked to share resources and ways to get involved. The first example I want to share is the Music and Memory Project that started with the movie and now has a great website with practical guides for clinicians and folks living out in the community on how to use music. Another program that I'm involved in, I'm actually on the board for, is actually based out of Minneapolis, but they have virtual choirs as well as in-person choirs for those who live in Minnesota, specifically looking and targeting those who are living with dementia. And then last but not least, the program that you may be more familiar with is local to Duke. At the National Museums, they have the Reflections Tour, which is an arts space, and that does now include also music aspects that has shown tremendous potential as well. And in interest of time, I'm going to skip the next couple of slides and end with these sort of parting words. In the last, I would say, 5 to 10 years, there's been also interest from the NIH and the Renee Fleming Foundation in sort of doing more work in this area and a really understanding of how music impacts the brains of those living with Alzheimer's disease. So part of my funding has come through that mechanism. And so it's really exciting to see where this work will take us. And I will just end. I'm almost out of time. These are being relatively new to Duke, I'm always interested in collaborations and working with others in this area, or how music can be used as a tool for other clinical populations, and how it can also involve the caregiver in the delivery, as well as how they can participate in music experiences as well. So those are some of my next steps, but I'll end here.
Thank you. It's very intriguing. I think we could take just a couple of questions here, and then, or we could take them at the end. If anyone has a burning question now, or comment, just wait a minute and see.
I have one question or thought. This idea that music is preserved in like these really later stages of Alzheimer's is interesting. I don't know if this is how you think of it, but I think of music as like a form of like semantic sort of memory. It's really been there for a very long time. And I'm wondering, what is it about music that makes it so different from other types of like semantic memories, like vocabulary and our memories for like world knowledge? Because it seems like there's something distinct about memory.
Yeah, and I think music. Yeah, when you think about also, one theory is that, music is often connected to important events, right? Part of our lives, so births, weddings, sad moments, right? Like losing people, like being at funerals. So I wonder if it is that, because I wonder if that is something about that particular emotional connection to music that has maybe a stronger effects later on in life. But that's just my speculation. But that's a really great point related to semantic memory. Thanks, Jenna.
It is very interesting. I know that there are some homeschool groups that actually teach some of their classes set to music. I don't have any data or anything about it. I'm just aware of it. And that apparently that seems to enhance their ability to recall later, the students.
Yes. I mean, we do that with, with our babies. For those of us who have little ones, we do this for mnemonic, right? Like I have done this, studying for exams or making up songs and kind of coming to them. So
yeah, it's the alphabet.
Yeah, exactly.
Have a little tune, but I'll spare you all. I can still remember it. Okay, well, listen, Dr. Petrovsky, that was really fascinating. Thank you so very much. In the interest of time, we'll move on to our second speaker. And at the end, if there are other questions after Dr. Buck takes questions, we can entertain those. So, Dr. Buck, please.
Sure.
All right. So as Jan said in her kind introduction, I am a neuroscientist by training who has really just recently kind of joined the Alzheimer's and related dementias field. I've, for the most part, been in my graduate and postdoc time researching Parkinson's disease, and I will get into through this talk how that connects to Alzheimer's and related dementias through my work. But so as a postdoc, I've been researching LRRK2 or leucine-rich repeat kinase 2, which is mutations in the gene for LRRK2 is responsible for, is one of the most common mutations responsible for autosomal dominant or familial forms of Parkinson's disease. And the most common mutation, as well as the other pathogenic mutations in LRRK2, increase its kinase activity, and the most common being G2019S. And so this LRRK2 kinase activity leads to autophosphorylation of the LRRK2 protein, but also phosphorylation of these Rab GTPases. And these Rab substrates of LRRK2, as well as LRRK2 itself, have been found to have diverse roles in the cell, from lysosomal and autophagosomal functions to mitochondria to immune functions. And while it's been established that these mutations in LRRK2 are causative for Parkinson's disease, the exact connection between these diverse roles of LRRK2 and the manifestation of Parkinson's has not really been thoroughly established. And so where my work has come in is investigating the connection between LRRK2 and tau pathology, which is slightly counterintuitive, as Parkinson's disease has been characterized by aggregation of a different protein, alpha-synuclein, in the form of Lewy bodies, while tau protein aggregation in the form of neurofibrillary tangles, as well as amyloid beta aggregation, are more canonical to Alzheimer's disease. But recent work has established that tau aggregation is actually more common in Parkinson's disease than was potentially previously thought. Here's just one image from a recent paper establishing this, where they looked at 4 cohorts of patients, those that were kind of control subjects with, elderly patients with no motor deficits, then kind of 2 cohorts of patients that were kind of considered to be potentially prodromal Parkinson's disease patients, where they had mild motor deficits but no definitive Parkinson's diagnosis, and they were split into those with Lewy bodies identified pathologically and those without. And then finally, they looked at diagnosed PD patients post-mortem as well. And this is looking at dopamine neurons, pigmented dopamine neurons in the substantia nigra, which is the brain region that, degenerates in Parkinson's disease and causes these motor symptoms associated with it. But in this figure here, you can see that in both Parkinson's disease patients, as well as these mild motor deficit patients, labeled in black here is phosphorylated tau protein. And you can see that there's aggregation of this tau protein in neurons in both PD patients, although it's much less so because a lot of these neurons have degenerated, but it's also very strong in these mild motor deficit patients, suggesting that tau, in addition to, Lewy bodies and synuclein, might be driving neurodegeneration in Parkinson's patients even potentially in patients that don't have Lewy body, which is occurring in a subset of Parkinson's patients. And so connecting that back to LRRK2, recent work has identified that tau pathology is very common in LRRK2 PD patients. And so this paper from Michael Henderson and Virginia Lee looked at 10 LRRK2 patients, PD patients with the most common mutation, G2019S. And surprisingly, a very solid amount of those patients did not have clear alpha-synuclein or phosphorylated alpha-synuclein labeling in the brain post-mortem and didn't have any clear identification of Lewy bodies. But even those patients and those that did have Lewy bodies had very strong labeling of phosphorylated tau, suggesting that tau is not just potentially a partner in neurodegeneration in LRRK2 PD, but might be able to drive it independently of alpha-synuclein. And this has been replicated to a limited extent in animal models of LRRK2 PD, where we see accumulation and transmission of this hyperphosphorylated tau that is elevated in the LRRK2 models compared to control animals or control models or control lines. But to date, there has really been no established mechanism connecting the elevated LRRK2 kinase activity seen in these pathogenic mutants to tau aggregation in Parkinson's disease or in LRRK2 Parkinson's disease. So where my work comes in is based around the hypothesis that LRRK2 kinase activity is indeed associated with tau in Parkinson's disease, and I also wanted to explore the possibility that it's associated with tau in other non-PD tauopathies like Alzheimer's and primary tauopathies as well. And so to begin investigating that potential connection, we've been working with Jerry Wang, who's the head of the neuropathology core here at the ADRC, and with him and the brain bank that he has been collecting samples through, we were able to obtain postmortem brain tissue from Alzheimer's patients, patients with dementia Lewy bodies, which is kind of on the spectrum between Alzheimer's and Parkinson's, where they have Lewy bodies, but also dementia and very commonly tau pathology, and then unaffected control subjects as well. And with those dementia with Lewy body patients, we were able to obtain subjects with both low and high tau pathology burdens to look at the difference between DLB subjects with Lewy bodies but without tau and those with tau as well. And then we performed immunohistochemical labeling of 2 LRRK2 kinase activity markers. Because again, LRRK2 phosphorylates these Rab proteins. And so we use established antibodies for phosphorylated Rab10 and phosphorylated Rab12, which are both LRRK2 substrates. And so theoretically higher, and LRRK2 is the predominant kinase of these 2 Rab proteins. So increased phosphorylation is a good correlate of increased LRRK2 kinase activity. And we looked in the hippocampus and temporal cortex where tau pathology should be high in subjects, as well as the frontal cortex that's kind of a regional control where there should be less tau pathology in these patients. And so strikingly, looking at PRAB12 first, we see that there was not much clear labeling in any region in control subjects that were age matched to these diseased patients. But then in Alzheimer's disease patients, it appeared that phospho-Rab12 was labeling tau aggregate structures, as it appeared to be labeling neurofibrillary tangles and dystrophic neurites and neurons across these brain regions. And consistent with that, in DLB patients with low tau, we did not see that form of labeling, although it did appear that phospho-Rab12 was labeling Lewy bodies, which are these nice, pretty, spheres that are just in a part of the cytoplasm of neurons. But then again, in DLB subjects with high tau, we see a mixture of both these tau-like structures or tangle and dystrophic neurite-like structures and Lewy body labeling by that phospho-Rab12, suggesting that it is localizing to both tau aggregates and Lewy bodies. And then when we quantify this, every group has significantly increased phospho-Rab12 signal compared to control subjects across the regions. And there was a regional difference where the labeling was higher in the hippocampus than the frontal cortex, consistent with that regional difference in pathology. And then consistent with this, that phospho-Rab12 is labeling tau, we looked at the correlations between that phospho-Rab12 signal and the different amyloid, tangle, and iridic plaque staging of these Alzheimer's control and DLB subjects postmortem. And we see that, for one thing, the labeling was positively correlated with all three stagings of Alzheimer's disease pathology, but the correlation was strongest with the tau tangle staging, again suggesting that it's labeling tau. And this wasn't observed for, or a positive correlation wasn't observed for age or postmortem interval. And so then, interestingly, something that we weren't really looking for, we found that that phospho-Rab10 and phospho-Rab12 were labeling this relatively less described pathology that I will explain in a minute after I go through the findings. But it looks like phospho-Rab10 and phospho-Rab12 are labeling granulovacuolar degeneration, which is associated with tau pathology, and I'll dig into that more deeply. But first, just to go through the findings, we saw that in both AD subjects and DLB subjects with high tau pathology, there was an increase in the number of neurons that we saw that had these puncta-like structures or granule-like structures labeled by phospho-Rab10 and phospho-Rab12. And that form of labeling is indicative of granulovacuolar degeneration. And quantifying, this was a significant increase in just the DLB patients with high tau for the phospho-Rab10, and then significantly increased in both Alzheimer's and DLB subjects with high tau in the hippocampus in phospho-Rab12. And we did not see very much labeling in the temporal cortex or the frontal cortex, but that's consistent with this type of degeneration that's specifically predominant in the hippocampus of tauopathy patients. And again, as we saw with just the total phospho-Rab12 signal, the level of this labeling of GVD correlated with, again, all stages. It wasn't particularly stronger for phospho-Rab10 in terms of correlating with the tangle stage compared to the other stagings. But for the phospho-Rab12, again, at least numerically, the correlation was strongest with that Brock tau tangle stage. And again, neither measure was correlated with age or postmortem interval. So what is granulovacuolar degeneration? These are granule-containing vacuoles that are found in the cytoplasm of neurons. They're either single or double membraned, and they have been shown to be, their presence is increased in Alzheimer's disease and primary tauopathy patients. And recent work, since these are very understudied, only recent work has really delved into the nature and identity of these structures, but they have been shown to be lysosomal structures that are part of the endolysosomal pathway. And lysosomes, contain cathepsins and other enzymes to break down proteins like tau. And but these GVD bodies are also positive for other kind of vesicular bodies along the endolysosomal pathway, although predominantly they are positive for lysosomal structures and not like autophagosomal markers. And importantly, these structures are proteolytically active, so they are still actively, have proteolysis activity and are able to break down proteins. And importantly, in the context of Alzheimer's disease pathology, their regional spread in Alzheimer's disease brains mirrors the spread of tau pathology, where they're particularly high in like the hippocampus and entorhinal cortex, and their spread is lower or later in other regions. And they have been shown definitively to be induced by pathological tau, but not amyloid beta in mouse models, and that's consistent with our labeling being correlated with tau pathology. But one thing that's interesting is that their appearance in neurons is associated specifically with early tau pathology, where at early stages of tau pathology, you see the appearance of these granulovacuolar degeneration bodies in a neuron. But then as that neuron develops into a more mature tau aggregation stage of mature neurofibrillary tangles, these GVD bodies tend to go away. And so it's really not understood why neurons might have GVD, but then once they develop formal or mature neurofibrillary tangles of tau aggregates, those bodies go away. And so the mechanism of their formation and their disappearance really is unknown, but people have hypothesized that it's related to this dysfunction and disruption in lysosomal, pathways and events. And it may be, in addition, people have hypothesized they may actually be protective and help potentially try to break down these tau aggregates, but then maybe they get overrun, or they might be contributing, these dysfunctional lysosomes potentially might be contributing to that degeneration as well. And so we found that phospho-Rab10 and phospho-Rab12 have labeled these GVD bodies as well as other tau and synuclein aggregates but we haven't really definitively, co-labeled those markers with those protein aggregates. And so we wanted to try to first see if we could get some sort of co-labeling that's definitive, but also try to replicate these findings in a mouse model to prove that this isn't some sort of artifact of, that's specific to the humans, and this is something that we can induce artificially in mice with, artificial, tau aggregation. And so to do that, we used a PS19 mouse model, which expressed the human P301S mutant tau in neurons. And this is a form of tau that, this is a mutant form of tau has been shown to be pathogenic for frontotemporal dementia. And it induces, just like in these frontotemporal dementia patients, it induces an age-dependent accumulation of hyperphosphorylated tau aggregation and induces neurodegeneration because of that. And so we looked at aged wild type and PS19 mice, and we labeled them for phospho-Rab12, phosphorylated tau by AT8, and then a GVD marker, casein kinase 1 delta or CK1 delta, to see if we could replicate that GVD as well. And so first, just confirming the model, we see that in the PS19 mice, in neurons of age PS19 mice, there's a stark increase in phosphorylated tau labeled by AT8 compared to essentially no labeling in wild type mice. And so we know that this model is working. And then, consistent with that GVD labeling in humans, we were able to find in the PS19 mice, specifically really in the entorhinal cortex, but also in the hippocampus, labeling of this CK1 delta positive GVD in neurons that was also positive for the phospho-Rab12. And the co-localization of these two markers was pretty stark, where the vast majority of CK1 delta positive GVD bodies were also positive for the phospho-Rab12. And then as that's shown in the quantification here, where we see GVD that is CK1 delta positive in the PS19 mice in both the hippocampus and entorhinal cortex. And then we see that these GVD bodies are almost completely positive for phospho-Rab12. And to confirm that they're lysosomal structures, we see that we use the marker LAMP1, which is a lysosomal membrane protein. And looking at a zoomed in image here, you can see that basically for GVDs that are positive for phospho-Rab12 and CK1 delta, LAMP1 kind of forms this ring around those two markers, as you can see here, suggesting that basically LAMP1 kind of labels the outer membrane of these GVD bodies, since this is a double membrane structure. And then the phospho-Rab12 and CK1 delta labels this more dense inner core of these GVD bodies, although it's not exactly established why these markers are labeling one membrane or structure of the GVD, but not the other. But nevertheless, this does show that these are lysosomal structures. And then interestingly, phospho-Rab12 labeling of these GVD bodies, one also partially co-localized with phosphorylated tau itself, suggesting that these GVD bodies might contain, tau, phosphorylated tau fragments, or might be trying to break them down. But then interestingly, again, showing that this phospho-Rab12 labeling is correlated with tau pathology, we see that in neurons that have very high levels of AT8 and might be like, pre-neurofibrillary tangles in these mice neurons, we see that in neurons with that higher labeling, we have more PRAP12 or phospho-Rab12 puncta, suggesting that the level of this phospho-Rab12 GVD labeling increases with tau pathology in these PS19 mouse neurons. And that is significant when we compare the number of phospho-Rab12 puncta in mice with high levels of AT8, or in neurons with high levels of AT8 compared to neurons with low levels of AT8 or phosphorylated tau. And so to summarize these findings, we see that in human neurodegenerative diseases, phospho-Rab10 labels these GVD bodies, and phospho-Rab12 labels Lewy bodies, tau aggregates, and GVD. And then in mice, we were able to replicate that PRAB12 labeling of GVD, and we were able to show definitively that it's triggered by pathogenic tau accumulation. And so what this suggests is that LRRK2 may contribute to pathological tau aggregation via this phospho-Rab associated GVD formation. And so our future directions are to, one, expand on these findings in humans by looking at Parkinson's disease and LRRK2 Parkinson's disease patients with lower high tau pathologies, and then to also determine the level of labeling in primary tauopathies as well. And then to double label these phospho-Rabs with tau, GVD, and synuclein markers to confirm that they are labeling these protein aggregates. But then in mice, we want to start to investigate that potential mechanism. Is LRRK2 kinase activity responsible for this phospho-Rab12 labeling of GVD? And conversely, if we inhibit LRRK2 in these mice, will we not just prevent the phospho-Rab12 labeling of GVD, but might we prevent the GVD, accumulation or appearance altogether? And similarly, we can do that by crossing LRRK2 knockout mice with these PS19 mice to see the level of GVD, as well as look at the level of tau pathology overall at these older time points in these mice. And so with that, I would like to thank the Sanders Lab and the Department of Neurology, who have helped me with all of these, experiments and getting this up and running, particularly Lori Sanders, my mentor, and Claudia Gonzalez Hunt, who is now a PI at Vanderbilt. And with that, I thank you for your time, and I will take any questions.
Thanks so much, Silas. It's awesome. I have a question I'll sneak in before it breath. So just in your final piece there about, thinking about the pathways towards the end, do you have any guesses? I mean, when I think of LRRK2, and again, it's not like my own personal special gene of interest, but it's one I've, come across enough. I think about like neuronal plasticity, autophagy, innate immunity, all of which certainly seem like they could be, a disruptable pathway underlying, some of these phenotypic consequences you're seeing. And so, do you have any guess or any hints or any kind of way to prioritize the ordering of looking into this?
Yeah, and it's definitely something interesting that I've been thinking about in terms of the cell type dependency, where a lot of those, findings with LRRK2 and immunity are in, astrocytes and microglia.
Yeah.
But, these GVD bodies are really only appearing in neurons. But there's this whole debate on how much LRRK2 is even expressed in neurons and which neurons at baseline and that kind of thing. And so it's hard to parse out, an actual mechanism, endogenously in, these mice that have very low levels of endogenous LRRK2. And so one thing that
but just real quick, are the microglia that they don't express LRRK2 typically, is that right? Or they do?
I mean, I can't remember microglia specifically.
I mean, but they're not forming the body, so I guess that's sort of, I'm just thinking about, LRRK2 expression is cell type specific, but is that cell type specificity the same across murine and human model, or murine and just people, right?
Yeah. And that has been explored to an extent, but we're kind of limited by tools at the moment, because it's not that LRRK2 is not expressed in neurons or, microglia, but it's just at very low levels that have been hard to detect with just current, antibodies through, IHC or Western blot.
Are there other questions or comments for Dr. Buck? This is really fascinating work. I think the tau part is really intriguing because we've gone after the amyloid with modest success. And it seems to me that the tau research may hold some promise there, either by itself or in tandem with work against the amyloid deposition. So I think this is interesting direction.
And there's definitely a growing body of evidence connecting, endolysosomal dysfunction and tau, aggregation and pathology. And so it's an interesting kind of confluence here between the LRRK2 and its known lysosomal roles, this formation of this kind of lysosomal pathology and that tau.